So now I like to introduce to our next speaker, Yuhan Hao. Yuhan is a PhD student at the New York University and a member of the Satija lab at the New York Genome Center. His research has focused on developing new computational tools for the analysis of single-cell genomics data, particularly for single-cell multimodal data. And he has developed the weighted nearest neighbors method to define cellular states by integrating information from multiple modalities. And that's what he's gonna talk us about today. Yuhan, the floor is yours.
Right, thanks for introduction. Can you hear me?
Yes.
Okay, cool. Right, so hello everyone. I'm Yuhan Hao, the PhD student from Rahul Satija's lab. I'm very excited to give a talk here. And today I'm going to talk about the integrated analysis of multimodal single cell data. And when we talk about the single cell, right, one of the key topics is always related to the cellular identity. So usually the cellular identity is defined on a single modality, such as morphology, functionality, surface proteins, or cellular transcriptome. But now these new multimodal techniques allow us to learn multiple aspects of a cell at the same time. So sometimes the expansion of knowledge may leave us more questions. The more aspects we know from a cell, the more uncertainty we have about the definition of cellular identity. So how do we define the cellular identity using those multiple sources of information? So here I will tell you several stories trying to convince you that our multimodal method can answer this question properly. So today, most of my stories will focus on CITEseq data that Eleni just introduced, which measures the RNA and the surface protein simultaneously for individual cells. Right? It adds a unique barcode to each antibody and also a polytail to the antibody through the disulfide bond. So it creatively converts the surface protein to the barcoded transcripts. And my first story is about a cord blood single cell CITEseq dataset. These are two typical matrices from CITEseq. So one is from RNA, another is from protein. You can see the RNA matrix here is very sparse, but the protein signal is very strong. But on the other hand, here we only have 10 antibodies, but we measured thousands of gene expressions. So you can see each data type has its distinct strengths and weakness. So here are the RNA and the protein UMaps for the dataset. You can see overall we get agreement between these two modalities. But let's have a look at the CD4 and the CD8 T cells here. Right, those two populations are connected with each other in the RNA space. And it seems that they had a continuous relation with each other. But in reality, they are very different cell types. But in the protein space, right, you can see the CD4 and the CD8 T cells are grouped very clearly. But however, in the protein data, we also lose certain cell types, such as dendritic cells, which are blended into the CD14 monocytes. I mean, it's not surprising as we didn't include the protein markers for the dendritic cells. So you can see each modality provides unequal information for individual cells. But how can we quantify or compare the information from these two modalities? So the basic strategy we used here is to predict the molecular content of a cell from its neighbors. So the intuition is that if neighbors of a cell from modality A cannot predict the molecular content from modality B, it means the modality A contains less information than modality B. So for example, the target blue cells showing in both panels is the same CD8 T cells. And in the RNA space, its neighbors are a mixture of CD4 and CD8 T cells. That's going to mess up the accuracy of protein prediction. While in the protein space, all the neighbors are CD8 T cells, so it can predict the RNA content correctly. So for the blue cell here, it should have higher protein weight. So here we are comparing the measured and the neighbor predicted molecular content of the blue target cell. So each dot here is a protein. So you can see for the RNA neighbors, right, it cannot predict the CD4 and the CD8 protein expression correctly, since the RNA neighbors are a mixture of CD4 and CD8 T cells. But from the other side, each dot here is a gene. You can see the protein neighbors can predict the RNA expression correctly. So based on this cross-modality prediction error, right, we can calculate the modality weights for all the cells. So this is the cell-specific modality weights for those different cell types. You can see for the CD8 T cells, right, it have a higher protein weights and low RNA weights. And but for the dendritic cells here, it have higher RNA weights because we know it doesn't have the markers in the protein panels. And also you may notice for some the mouse cells here used as a negative control, as a negative protein control. So it also have a higher protein weights. It fits our expectation. So you can see using this modality weight, we can construct a new weighted distance matrix, which can extract the important information from both modalities. And using this weighted distance matrix, we can define a weighted nearness neighbor graph that can be used for all the downstream analysis. So this is multimodal representations of the cord blood dataset. Right here, we perfectly separate the CD4 and the CD8 T cells and also recover the dendritic cells. And interestingly, the NK cells are divided into two groups, CD8 positive and CD8 negative. So it is we have not observed before using the single-cell RNA-seq alone. So you can see our methods take the benefits from both RNA and the protein to get a better cellular identity. And let's see the second story about another CITEseq dataset from the bone marrow. It contains 30,000 cells and 25 protein markers. In the bone marrow, there are prepotent progenitor cells and also the differentiated immune cells. And our protein panels mainly contains the markers for the differentiated lymphoid populations, but not for those different lineages progenitor populations. So these are the 2U map for the bone marrow data set. And you can see in the RNA space, right, those T cell populations cannot be effectively separated by the single cell RNA-seq alone, just because of the limitation of the technology itself. But in the protein space, you can see for those progenitor lineages populations, they are blended together as we expected. But let's see how our methods work in this case. First, you can see the T cell groups all receive the high protein weights. And for each of the populations with the highest RNA weights are those progenitor populations. This is consistent with the composition of our protein panels, which didn't contain the markers for different lineages of progenitors. And as a result, our multimodal analysis is able to identify the diverse progenitors and all those T cell populations. So this result suggests that our analysis can allow one data type to compensate for the weakness in another. Besides the CITEseq, we can also apply our WNN methods to the text multiome PBMC dataset, which measures the RNA and the chromatin accessibility simultaneously. So you can see in our multimodal clustering methods, it provides better cell clusters than the individual modalities. For example, the Treg populations separate better in the RNA space, and the CD4 and the CD naive populations have better separation in the ATAC space. And in our WNN representations, it keeps the advantages from both modalities. And the better cellular clusters will facilitate us to get stronger signal for the downstream analysis. But let's look at a more interesting CITEseq dataset. In collaboration with Peter and Stephanie from New York Genome Center Innovation Lab, we performed a CITEseq for 200,000 PBMCs with over 200 antibodies. And our multimodal methods can be easily applied to this large-scale dataset. So these are the RNA and the protein UMAPs. There's also a general agreement between these two representations. But this is the multimodal representations of the PBMC. What really stands out about this representation is that it shows so many clear cell populations, but also keep some continuity between the cell types. And we identified 57 clusters in this multimodal analysis containing all the major and the minor immune cell types, and revealing striking cellular diversity, particularly within these lymphoid lineages. And it gives us opportunities to define a multimodal atlas of the human immune system. And due to the time limitation, I cannot talk more detailed information about the clusters and this atlas. But right now, here, we have a good reference for the visualization. But what else can we do for this reference? And our motivation is that if we can use it in a similar way like the human genome, or which can be used to map new reads, can we map new cells into our reference? And let's go to the second part of my talk. A single-cell transcriptome profiling of the immune system can become routine, and not just for the healthy subjects, but also in clinical contexts, including the patients with COVID-19. So for example, this is PBMC RNA UMaps from a COVID-19 project from Catherine Blish group, and the data is processed by the unsupervised clustering analysis, and the cell annotations are added manually. However, our multimodal reference gives us a chance to do some supervised analysis for the additional single-cell dataset from PBMC. And our goal is to map new query cells to our reference UMap and also transfer the cell type annotations automatically. But the first question is that how do we map the single-cell RNA data to our multimodal reference? And in order to solve these questions, we first apply a procedure called supervised principal component analysis to the transcriptome data and in our reference dataset. Instead of seeking to identify low-dimension projections that maximize the total variance, as in PCA, the supervised PCA identifies a transformation of the RNA dataset that can maximally capture the structures defined in the multimodal graph here. So I will go through these methods, what is SPCA is, quickly. But the objective function of this SPCA is the Hilbert-Schmidt independent criteria, which measures the dependence of two representations. So one representation is a kind of unknown kernel from the RNA, and another L represents another kernel of the multimodal graph. And the goal is to find a linear transformations of RNA, a U here, that can maximize this H-seq value between these two representations and these two kernels. So fortunately, this question has a closed form solutions that is the eigen decomposition of this matrix. But generally, the SPCA allows the WN graph to help supervise analysis of gene expression data and identify a subspace of the transcriptome, a U here, that can maximally capture the structures in the WN graph. And when we have this U, this subspace, we can map our new query cells to this SPCA space and then do the analysis with our multimodal reference. So this is the mapping result for the COVID-19 PBMC dataset. The left one is from the UMap from our PBMC reference, and the right one is from the new query, the COVID-19 cells. And we keep the query UMap similar with the reference, and because it can provide a lot of convenience for us to understand the new query cells. And we can also find the same cell types in a similar UMap positions. But you may ask, are those cells mapped correctly? Let me show you the markers for two hard identified cell types. So all cells in the UMaps are the COVID-19 dataset. So RTKN2, a Treg canonical markers, is enriched in the query cells in this position, where the reference Tregs are located. And also the cyclic 6 is a canonical markers for the ASDC, is also enriched in the position where the reference ASDC cells are located. And I just want to emphasize that those query cells can correctly mapped to our multimodal reference UMAP in terms of gene expression. And we did some systematically validations in our preprint, and not just to map the UMAP. We can also transfer the cell type annotations from our reference dataset. And you can see here, these are the COVID-19 PBMC cells split by the healthy and the COVID-19. So it is clear that some cell types have different proportional change. For example, the expanded CD14 monocytes, the expanded plasmablast, and the depleted mate cells. And we also validate the depletion of mate cells using new healthy and COVID-19 samples by CITEseq. But the thing is that given the cellular identity for all the cells, we can do the proportional change analysis and the transcriptome differential analysis for each individual cell types between the healthy and disease easily. So it just provides a lot of convenience to study the complex immune system. And we also created a web app called Asmus, and you can upload your count matrix to this website. It will map your cells to our reference and transfer the cell type information. In the end, you can download your annotated object, and this app is also scalable pretty well. For the COVID-19 dataset containing 44,000 cells, it takes around 5 minutes to get the projected UMAP and the cell type annotations. So, okay, so to summary, and our weighted nearness neighbor graph represent the informations for two modalities. And it is single representations to define the cellular identity. And our WM methods also can be generalized to other multimodal data, such as text multiome, can also be used in the ASAP-seq and dogma-seq that Eleni introduced, and also can be applied for large-scale dataset. And we also created a web app, Asmus, can mapping the query single-cell dataset to our multimodal reference. And also you can use Sera to do this locally. And this kind of a mapping strategy have facilitated the interpretation of the complex immune system. And this method is implemented in the Sera v4. You can find the corresponding variants in our website. And if you have interested, you can try it in Sera v4 package. And if you want to know more detailed information about our methods, you can read our preprint in the bioarchive. So in the acknowledgement, first I have to thank Rahul for his insightful ideas and the guidance to make this project possible. And thanks for Peter and Stephanie from the Innovation Lab. They performed all those related experiments. And thanks for all other lab members for their endless help and the useful suggestions. So thank you for listening. Okay.
Thank you, Yuhan, sorry. So we have a couple of questions here on the chat box. So the first one is from Sergey Kolchenko. If you can unmute yourself.
Yes, thank you. So in one of the formulas you wrote, we saw that the distance between two cells is a weight and distance based on RNA content and measured proteins. And that allows you basically to have a better representation. But how is it different if you just take RNA measurements, protein measurements, combine them in one bigger feature matrix and make a UMAP projection? So what's the problem with that approach?
Yes, thanks for your questions. And the point is that I think the question you asked, right, if we just concatenate all those protein features into the genes and then apply the PCA or do those all kind of single modality, right? So I think the benefits of our multimodal analysis is get the benefits from the modality weights. Because you can think if your protein modality or if your antibodies kind of do not have a lot of noisy data, if you just concatenate them with the genes together, it will mess up the single modality analysis. But in our strategy, we will first to quantify the information content there. If the one data type, the quality is bad, it will have kind of a lower modality weights, and it will have less influence in the final cellular identity. And in our preprint, we tried to add a lot of noise kind of background in our protein data set. And we show that when with the increase of noise, the protein weights kind of goes down, and it can just keep the information from the RNA. And also, if the protein not good, it will not impair the entire analysis. So yeah.
All right, I see. Thank you.
Yeah.
Next question from JJ.
Hello. Hi. Yes.
Hello.
Thanks, Yuhan. Thanks, Sandra. Very interesting talk. I think it's fair to say that it looked like the myeloid subsets still struggled a little bit to be resolved from one another, even using the multimodal WNN. I just wondered, is this a focused RNA panel that you've used here, or is this whole transcriptional profiling? And what's your view on perhaps increasing the power maybe of these difficult to resolve populations by perhaps doing deep sequencing on a focused panel of, say, with a myeloid bias in combination with cell surface staining? They are notoriously difficult to pull apart. And I just wondered what you thought about sort of balancing deep sequencing with focused panels of RNA versus adding in more and more antibodies.
Yeah, I think it really depends if you have very good or specific antibodies that you have for the lineages or for the population you have. And if you have that, definitely, I mean, when you add those antibodies, it will dramatically change the cellular identity, or you can find those populations very easily. But if you don't have that, I think when you increase the sequence steps of the RNA, it can help, but it will not help a lot. For example, in those T cell populations here, for the CD4, CD8 naive T cells, and for those different T cell subpopulations, they are just very hard to separate in the single cell RNA-seq alone. Even though you sequence much deeper, kind of more deeper, it will not kind of separate them very clearly like the antibody parts. So I think it really depends on if you have the good antibodies for the populations you have interests. Yeah.
Thanks, Yuhan. Yeah, maybe that's where some of the chromatin comes in as well, and maybe you could add another layer on top.
Yes, exactly.
Yeah, cool. Thank you.
Thank you.
Next question from Austin Hopland.
Hi, Yuhan. Great
Hello.
talk. Really was looking forward to it and enjoyed it. I am thinking more about sort of like the whole embryo sort of approach coming from a developmental biology background. And I was wondering, in these approaches where we have sort of markers of many, many different cell types that may be like established well into the field, such like your antibody panel for the PBMCs. Could you imagine sort of like a manual curation of a database and sort of like a confidence that this marker really should be weighted more rather than just this sort of computational unbiased approach? Do you think there is an opportunity to add in more bias, I guess, but meaningfully?
Yeah, that's very good questions. So do you mean in terms of protein, or also even in terms of the RNA?
Specifically in protein, because I feel like that's still sort of the challenge, right? Is getting many proteins, whereas RNA sort of is the basis of a lot of these methods. It's sort of like the ground framework.
Yeah, cool. Right. I think in terms of the protein, I mean in any kind of the protein methods, if you use antibodies, right? So that will depend on two things. Is if your sample quality is high quality, if the cells have a high viabilities, because if the cell viability is low, even though you have those specific, you have some strong protein markers, it may not stain in the cells very well. So right, we can kind of put higher weights on protein or something. But you don't want to kind of split the populations by individual protein, right? You also want those populations, some markers, and you can also capture some transcriptome signature or some stuff. And in our methods, we kind of trying to eliminate that we took too much weights on the protein. We don't want the cells, for example, for those different T cell populations, they are all kind of get small isolated populations just because of the one protein. So that's why we think right now, even though it's kind of the unsupervised analysis, but the protein signal is very strong. If it has a signal, so it's already it's good enough. Right now to have this kind of the modality weights for those parts. Yeah, but
I was just imagining like embryo scale, you might need some greater separation, right? Because here we're looking at blood and immune function, but when we scale this out to, different, completely different lineages, that might be useful. But thank you.
Yes, I agree. Right. Thank you.
Next question from Yang Li. Yang Li, can you unmute yourself? Okay, in the meantime, I'll call on Gabriel Sassi.
Yeah. Hello, Yuhan.
Hello.
Thanks for the talk, very interesting.
Thanks.
I have a question about large data set for CITEseq. So sometimes you have to run CITEseq in different batches, in different time and in different experiments. And do you struggle in doing batch effect correction, especially because you have RNA and also the protein? So with Harmony, for instance, or CCA, you can correct for the RNA, and how do you do it for CITEseq and for the protein?
Yes, thanks. Right. So in our this large scale PBMC CITEseq dataset, we also have very strong batch effect. If while you work on the protein, you will see stronger batch effect because the antibodies may have different clone, and also those antibody staining have just more sensitive to different batches. And what we used is that we also use the CCA or the RPCA that in CERAV3, we can use that methods to integrate the protein side. So we correct the batch effect in the RNA modality, and we correct the batch effect, the protein, and then we use this WN methods to generate this multimodal stuff. But we are also working on a kind of multimodal anchors. If you are familiar with our methods, you know that we are using some anchors and to get rid of the batch effect. But in the way that I just introduced, right, we correct the RNA batch effect based on the RNA anchors and the protein by protein anchors. And we are working on just to use one set of anchor cells and to correct both RNA and the protein, right? So generally, right now, you can use the CCA or RPCA to correct the protein batch effect as well. Yeah.
Thank you very much.
Thanks.
Okay, so last question from Fiona Kerlin.
Hello, very fascinating
Hello.
talk. Since you said that WNN also works on ASAP-seq and dogma-seq, does that mean that it can integrate more than 2 data sets at once, like RNA, attack, and protein? And is there a limit on how many it can handle at the same time?
Yeah, it's a good question, right? In the dogma-seq, right, it has 3 modalities: the ATAC, RNA, and the surface proteins. Yes, our WN methods can work for the 3 modalities and even with more modalities. Because the main stuff we use this cross modality predictions. I mean, when you have more modalities, we just need to calculate some actual cross modalities prediction stuff. And then it can convert to the weighted distance matrix. So right now here, right, in our new defined weighted distance matrix, right now it have two modalities. When you have more modalities, just add some more to have a new weighted distance matrix. So yes, it can be applied to more than two modalities. And is there a limitation? How many WN, how many modalities? Right now, I mean, except, right, we can sequence the modalities to 100 or 1000, then the pairwise cross-modality prediction may not work well. But at least right now, right, we have 2, 3, 4, 6. I think this cross-prediction methods still can be scaled for those kind of methods, technology. Yeah.
Very exciting. Thank you.
Thank you.